Here we are today for another lab chat, and today we have Chip Petricoin from George Mason University here to tell us about his work using Arcturus LCM. Chip, can you tell me a little bit about your laboratory and the work that you're doing?
Well, thank you very much. Our laboratory is the Center for Applied Proteomics and Molecular Medicine here at George Mason University. We work on a lot of various diseases, most notably cancer and infectious diseases, diseases of neurodegenerative disorders, but largely we're an oncology-focused laboratory. And my colleague Lance Leota was one of the inventors of laser capture microdissection at the NIH.
I see. And I understand you have a recent cancer research study looking at glioblastoma that used LCM in a particularly unique way. Can you tell me a little bit about that?
Sure. So we tested the underpinning hypothesis of whether or not the unknown and unknowable amounts of stroma and other cells in a clinical tumor specimen affect the final proteomic and phosphoproteomic signature of those tumors. And we used a nice study set of glioblastoma tumors that were part of the Cancer Genome Atlas project's GBM tumor set. And we did so because, well, all of the TCGA data that's currently being developed is all done with ground-up tissue, none of it laser capture microdissected. And so we wanted to see whether or not this upfront sample preparation step made any difference in the final output.
Because at TCGA, the glioblastoma samples, there are 90% sort of tumor. But it's that 10% sort of you're looking at what kind of signals are contaminating.
Yes, I mean, the context of the TCGA data has always been, well, most of these tumors are fairly pure. They mostly contain the tumor epithelium, or in the case of glioblastoma, the tumor cells. And largely they are very high purity. However, you're probably in the 60 to 90% range in general with this set. So they're already pretty highly enriched, but we wanted to see, is this okay? You don't have to worry about microdissecting when there's this much high percent tumor. You know, it could be pretty logical for people starting out with, you know, tumor biopsies where they're, you know, 5, 10, 15% tumor. Yeah, probably would need to microdissect there because the majority of the bulk tissue is not tumor. It may be that once you start to get 60, 70, 80, 90% tumor, you're okay. And that's what we wanted to try to test.
And what did you find?
Well, quite surprisingly, even tumors that were 90% tumor, the phosphoprotein and protein measurements that were obtained were probably not as accurate as you might expect. What we did in our study is compared the microdissected to non-microdissected material in the exact same tissue specimen. We actually took the tumor and we bread loafed it and took part of it for microdissection and this part for non-microdissection, so there was no bias in the underpinning tumor content. And we arrayed these at the same time and then directly compared. What we found is out of the 40 cases that we looked at, only 1 out of the 40 actually looked like the microdissected counterpart.
Wow.
And then we were able to kind of prove that the laser capture enriched content was accurate because we were able to compare the protein data to the underpinning genomic data. It's like P10 loss and EGFR mutation. The microdissected material was highly statistically concordant with the expected genomic derangements, which were often entirely lost in the undissected material. So we were able to prove definitively that if you did not microdissect, even tumors that were fairly highly purified initially, you would get wrong information.
Wow.
We're not saying that all the TCGA data is wrong. We're just saying that you don't know what's right or wrong if your initial data set is contaminated by stromal cell populations, and you're not going to know whether or not that's going to impact your downstream analysis. So LCM is a critical component of everything we do. We go the extra length to make sure that the data is as accurate as possible.
Great. And as far as use of the Arcturus platform, what is it about the Arcturus that was critical as far as the kind of work that you're doing?
Well, as you may know, but it's perhaps lost on people that weren't part of the development process for laser capture, is that the underpinning patents for laser capture are to directly procure the cells from the tissue specimen. See the cells, pick the cells up. To get around that kind of intellectual property, other platforms had to be developed, such as was developed for Leica or Palm instruments, where you have to ablate the tumor and then catapult it. The Arcturus instrument is the only instrument that has the infrared laser beam, the IR laser. That IR laser allows the direct capture. UV lasers, which also the Arcturus instrument has both UV and IR, the UV lasers are high power. They ablate the tissue, they fry it basically, and this destroys the proteins, the DNA, and the RNA. A lot of people don't realize that high energy UV cutting, you cut, but then there's a field around it that the macromolecules are destroyed. The IR is a soft laser. We've published many papers where the IR laser has no effect on protein, phosphoprotein, any post-translational modification. And it's the only laser that allows you to directly pick it up, pick the cells up that you see. And that's the IR laser is really the dominating laser for molecular profiling, because RNA, DNA, phosphoproteins are completely unaffected by that laser, which is not the case for UV. Many people think, well, I'm just going to do microdissection of my tissue, and they'll come back to us and say, well, I don't see any protein. You guys are publishing all these papers on proteomics from LCM, and our first question is, well, did you use the Arcturus instrument or did you use somebody else's? Because the Arcturus instrument is the one you need to use for phosphoprotein and protein analysis, especially where you have small numbers of cells and they're very discrete.
Sure. Well, thank you, Chip, for sharing this morning.
Sure.
It's been great.
My pleasure.
If any of you have questions for Chip, simply use the LabChat hashtag over Twitter. And if you're interested in any of our other applications of the Arcturus LCM, you can access a video right here for further information via video format. If you're interested in other information about Arcturus, you can also access it at lifetechnologies.com/Arcturus. Lastly, if you're interested in doing a lab chat with us, feel free to contact us using any of our social channels. That is a wrap.